Hello, and how are you? Welcome to our session on cellular transport mechanisms. My name is Winnie Barawa. Keep it here with me till the end. And if you're new, I always say welcome to our channel where we handle content and lectures, lessons, and even interviews pertaining to healthcare. If this is your area of interest or your area of liking, remember to subscribe, give us a thumbs up, and click on the notification button so that you can be notified when we post our next sessions. So let's keep up with today's class on cellular transport mechanisms. We had discussed the cell membrane and we described it actually that it is semi-permeable, meaning it allows materials to cross in and out of the cell. However, we didn't go into details how the materials cross the membrane, and in today's class, that is what we are going to look into, how several materials are able to cross in and out of the cell, across the cell membrane, but again, we will look at other mechanisms that do not necessarily use the cell membrane, but they are part of mechanisms or mechanisms of transport. So this is what we are going to focus on. We are going to look at two categories of transport mechanisms. And the first one is what we call passive transport, and the second one we call it active transport. By definition, passive transport is transportation that does not use energy, while active transport we mean transportation mechanism that needs the use of energy. And in this case, the energy we are referring to is the ATP, adenosine triphosphate, which normally gets broken down to release energy and get converted into adenosine diphosphate. With the passive transport where there's no use of energy, then here we have categories such as diffusion, osmosis, and all that. So our key focus in this class will be diffusion, osmosis, facilitated diffusion, active transport, pinocytosis, phagocytosis, so let's begin with the first mechanism that we call diffusion. By definition, diffusion is the movement of molecules from area of great concentration to an area of lesser concentration. So, and in our diagram here, there is a demonstration of a diagram which has high concentration to low concentration. And with high concentration, we mean if it's an aqueous solution, we look at which solution is highly concentrated with concentrates or substances and which aqueous solution is not. Areas of application for diffusion can happen in the respiratory system where there's exchange of gases in the lungs and other body tissues. And during respiration, mostly this takes place around the alveoli where gaseous exchange takes place, and high concentration of oxygen from the atmosphere is able to diffuse into the blood system, while high concentration of carbon dioxide, which is in the blood, is able to diffuse out into the lung spaces and being taken away during the expiration or during the breathing out. In the body tissues, also in the respiratory functions, what we call internal respiration, we have blood which is highly concentrated with carbon dioxide, diffusing it out into the channels to take it out towards the lungs, while cells that has low supply of oxygen is able to diffuse in oxygen and be able to take it for metabolic functions. The second module is the osmosis, and with osmosis here we mean movement of water from region of greater concentration to region of low concentration. In other words, we call osmosis the diffusion of water. So this is movement of water from greater concentration to region of low concentration. In simple terms, it is defined as the diffusion of water. So with osmosis, the biggest focus is the water and not just any other solution. So with this demonstration here, you can see there's pure water on one side. that is targeting the other side of the solution, which has less amount of water. So for this process to happen, you need water on one side that is highly concentrated and needs to cross a membrane and head to the other side. So a process where water is highly concentrated on one side, moving to a region of low concentration of the other side, then we describe this as osmosis. In application, osmosis happens in areas that needs absorption. For example, strongly happening in the intestines, both small and large intestines. It also happens strongly in the kidneys where there is need for osmosis because of water retention and water elimination for the purpose of urine. Keeping on, we have what we call facilitated diffusion. And with facilitated diffusion, it's a type of diffusion, but a little bit different, because with the facilitated diffusion, we are using what we call transporters or carrier enzymes. And these are found on the channel proteins, which are part of the cell membrane. Just to remind us, under the cell membrane, where we were discussing cell membrane proteins, we had transporters or channel proteins that are part of the cell membrane. And what they do is they facilitate transportation of substances in and out of the cell membrane. So with the channel proteins, what they do is they are able to act as carriers or transporters, and so they take in materials that cannot diffuse by themselves. So for example, with diffusion, materials just cross the cell membrane on their own without the need of transporters. But now when we are doing facilitated diffusion, when we need to take in these materials, we will need carrier proteins. So what happens is with our carrier proteins, where we're talking about the carrier proteins, the materials will come, attach on the receptor sites or attach on the active sites and cross to either in or out of the cell. And because this is an assisted, it's more like an assisted transport, we then call it facilitated diffusion. However, the same thing applies about diffusion here. The materials need to be moving from a region of high concentration to a region of low concentration. So if you're observing our insoluble molecules on this diagram, they are more on one side, and that is our extracellular fluid, and they are less in the cytoplasm. So to allow these materials to cross, because they are a little bit huge, they can't pass on the cell membrane or they can't pass across the cell membrane, so they'll get attached on our carrier protein here and they will cross into inside the cell. So most of the cell take in glucose by facilitated diffusion, or we are being told the large molecules of glucose which can't diffuse are most of the time facilitated using facilitated diffusion. And for this, it means they get attached on the carrier proteins or transporters, and they can be crossed over into the other side of the cell that is needed. And the rule is they need to be moving from a region of high concentration to a region of low concentration. Active transport. Active transport is our fourth mechanism, and here we describe active transport as a movement whereby cell uses ATP. ATP is a form of energy to move substances from an area of lesser concentration to an area of greater concentration. So this is a movement from low to high concentration. So we are told that for this to take place, you need to use energy. And the energy we apply here is the ATP, adenosine triphosphate, that will be broken down into ADP. So where does this active transport happen most of the time? It can happen on the nerve cells and muscle cells, where we end up using something we call the sodium-potassium pumps that will allow sodium ions to the exterior of the cell and also allow potassium ions into the cell. And while this exchange of sodium potassium is happening, it's being able to facilitate the nervous stimulation and the communication of the cell across the nerve cell. So when this is happening, it needs to use energy, and because it is using energy to pass the ions in and out of the cell, then we are describing this as active transport. We are also being told that small intestines are able to absorb glucose and amino acids Most of it is done by active transport, or rather it's being facilitated by the energy. But in this, we also use transporters, or we use the channel proteins that helps us to establish the active transportation of the substances. So in simple term here, we mean active transport means transportation or movement of substances from low concentration to areas of greater concentration. And this, as you can observe, it is against the gradient, because the normal gradient is high concentration. It is high concentration to low concentration. But the active transport is moving from less concentration to high concentration. Therefore, it is against the gradient. It is like you're climbing a mountain. And so to keep up with this, you need energy. And we have discussed where do we find this active transport mostly in. You can find it in the nervous cell, you can find it in the digestive system, or in areas where there's need of energy to transport materials. So let's look at the differences. We've said transport mechanisms can be passive or active. And with active transport is what we've already described, that this is movement from a region of low concentration to high concentration using energy or against the gradient. So we'll be using energy here. While in passive transport, we are saying this is movement of materials without the use of energy. And it can encompass mechanisms including diffusion, what we simply call simple diffusion. It can include facilitated diffusion, and also it can include osmosis. So these are mechanisms of transportation across the cell membrane. And the difference is one needs energy while the other does not need the use of energy. And passive transport can be diffusion and osmosis. Active transport will be part of the main active side of using energy, and that is just what we call active transport. There isn't like a specific name for that. Next type of transportation, what we call filtration. So the biggest difference with filtration as a mechanism is that it uses pressure as opposed to the others where we talk about using energy. Filtration uses pressure. So we are saying this is a mechanism whereby pressure forces water and dissolve materials through a membrane from an area of high pressure to an area of low pressure. So this is a mechanism that is forcing water and dissolved materials through a membrane from an area of high pressure to an area of low pressure. Good example is issues like tissue fluid, which is said to be formed mainly by filtration. And this happens because blood pressure most of the time can force plasma and dissolved nutrients out of the capillaries into the tissue spaces. And so because of the high pressure in the blood vessels or in the areas in the body, partly it filters out materials, and this can include plasma part of the blood, and it can also include dissolved nutrients of the body system. And these nutrients can be pushed out through the capillaries, and they can be taken into the tissue spaces. And this fluid is what we call the tissue fluid. So we're being told that application, or where we find filtration, we find it mostly in the kidneys because of the process of urine formation. And to form urine, we need filtration to filter out excess water, to filter out unnecessary ammonium or ammonium salts that are needed in the body, are not needed in the body, and so can be eliminated as waste materials in the urine. So this happens in the kidneys actively. That's how filtration works, from a space of high pressure to an area of low pressure. And it gets applied mostly in the kidneys during the creation of urine. And we have the other part of mechanism that we call endocytosis. So endocytosis as a mechanism of transportation, this is where we mean substances are taken into the cytoplasm from the outside part of the cell. So this is a process whereby substances are taken into the cell from the external part of the cell, what we call extracellular, into the cytoplasm. So this process of endocytosis is facilitated by vacuoles or vesicles. And so what happens is the vacuole or the vesicle will come, attach on the cell membrane, open and take in substances. Then once they have successfully done that, they pinch off from the cell membrane and they move inside the cell to supply them to the needed areas. So the process uses two main mechanisms, what we call phagocytosis and pinocytosis. These have two different descriptions. Phagocytosis, we are saying this is a form of endocytosis, but it allows a moving cell to engulf substances, and this could include dead materials, dead cells. It could include toxic substances with the purpose of destroying them. So this is the engulfing of substances into the cell for the purpose of destroying them. While the other side of pinocytosis, it's another form of endocytosis, but this one entails engulfing of molecules for the purpose of reabsorption or metabolic processes. So pinocytosis does not target to destroy. Pinocytosis targets for the purpose of reabsorption or for the purpose of enhancing metabolic processes. So it engulfs materials with the main reason of reabsorption or metabolism. So what is the difference? So where do we find phagocytosis? We are saying phagocytosis will destroy or engulf for the purpose of destroying, and so therefore you will find it being established mostly by the white blood cells, because these are the ones that carry out destructions of microorganisms, or it also helps in destroying pathogens in the body. While pinocytosis, you can find them in the kidney tubule cells, where there is need of transporting proteins, where there's also need of eliminating extra amino acids or extra ammonium salts that are not needed by the body. So here is an outlook of endocytosis. So we are saying it can happen in the format of phagocytosis, or it can happen in the format of pinocytosis. So with phagocytosis, we are mainly focusing on engulfing materials for the purpose of destroying them. So we engulf a microorganism, for example, here, and we bring it in a vacuole. And so the vacuole will then be destroyed either by attaching a lysosome into it, pouring the lytic enzyme inside here and destroying it. But when it comes to the pinocytosis, we bring in the substances, but the purpose is to absorb the substances. We are not targeting on destroying the substance. And so it will be engulfed, pinch off in the vesicle or the vacuoles, and then it can be taken into the cell for other reasons. There are other types of endocytosis, what we call receptor-mediated endocytosis, which means for the materials to be taken in, there is need for the signal receptors to signify which materials to be taken in. And so when the substances will be coming into the cell, they will first attach to the receptor sites, as you can see here. And once they are attached to the receptor sites, they'll therefore be engulfed, or they will be encircled, and then they will pinch off and enter the cell. So this means these are materials that have to be facilitated or have to be mediated into the cell by the use of receptor sites. So it's a bit different as just doing phagocytosis or pinocytosis. And the last mechanism that I thought it's wise to include in our video today is the exocytosis. And exocytosis, it's a process of transporting materials from inside of the cell to outside of the cell. And we are being explained that exocytosis uses energy and it is considered to be part of active transport. So by description or illustration is exocytosis is whereby you want materials to be emptied out of the cell. And here we will still use the vesicles or the vacuoles. And so the newly formed vesicles which have engulfed our materials here will move towards the cell membrane. It will attach, or it will fuse here. After fusing, it will open up and then release our materials outside. So this is simply what we mean by exocytosis. The process of transporting materials from inside of the cell to outside of the cell uses energy, and sometimes we consider it as part of active transport mechanisms. So this is the difference between exocytosis and endocytosis. Exocytosis, we are saying materials are being transported from inside of the cell towards outside of the cell. So they are being removed from the cell. While endocytosis, we are taking in materials from outside of the cell into the inner side of the cell. And the main work that is happening here is we are using vacuoles and vesicles. So they will all be coming to the cell membrane. At the endocytosis, we are picking them at the cell membrane and we are pinching off and going inside the cell. Exocytosis, the vacuoles or the vesicles will be moving to the cell membrane, attach and empty the products out of the cell. So that is the difference between exocytosis and endocytosis. They are all mechanisms of transportation with the purpose of facilitating movement of materials in and out of the cell across the cell membrane. So thank you so much for being with us to the end. Again, if you've not yet subscribed, please build up our family of learners on this channel by subscribing, and also click on the bell sound so that you can be notified when a new video or a new class has been scheduled or been posted for you to enjoy and share with your community of friends or learners, and like the videos. So just to remind you, my name is Winnie Barawa again, and see you in our next class. Bye bye.